Okay, so the second of our four major biomolecules is protein. And we did protein synthesis in biology, and we talked about how DNA carries a code, and mRNA is copied from that code and goes out into the cytoplasm of the cell, where the ribosome can then read the mRNA 3 nucleotides at a time, called a codon. tRNA brings in the correct amino acid, it's linked together, and then it's released into the protein. And that is true. It's a very simplified version, so we're going to learn it a little bit deeper this time. When you're looking at a molecule, the nitrogen is what helps you to determine that you're looking at a protein. They also have what we call R groups, but nitrogen is one of the key things that you're going to look for, key for recognition. So proteins is the most abundant organic molecule in the body, something that contains carbon. So it's the most abundant. That you are thousands of different types of proteins just working together and doing their specific jobs. So protein monomers, when we talked about this in biology, we talked about how amino acids are linked together, one amino acid at a time, very specifically based on a codon. Now, I'm not going to make you remember or learn or practice how to read the codon chart, but if you all remember, there was a wheel, and you have to go and you like A, U, G, and then you find that that codes for methionine or whatever. So, but all of your proteins, all of your thousands of different types of proteins are made of only 20 naturally occurring amino acids. We only have 20 that make up thousands of different types of functioning proteins, whether it's from an enzyme that speeds up chemical reactions, which you have lots of different types of those, just to a carrier across the cell membrane or whatever. You have thousands of different types of proteins that you could not function, you would not work without those. So something good is almost all 20 amino acids are present in every single protein. So when you eat, the reason you need to eat a higher protein diet, didn't mean to hit the camera, if you're trying to gain muscle or something like that, is because those are highly concentrated in protein, and when you eat another protein, you break it down. You break down, you catabolize that large protein into its monomers, which are amino acids, and then you take those amino acids and you build your proteins from that. And then there are 8 essential amino acids that we can't create, our body can take, because, these amino acids are made of our SNOPS elements and stuff, so, your body can sometimes make them, but there are 8 that we cannot make. You have to take them in your diet. So, it's very important. A lot of malnourished people may not necessarily look super thin or whatever, but they might be malnourished, and that's because they're not getting the essential elements or the essential monomers that they need from their food. If you don't eat a balanced diet of carbs and proteins and fats, and of course you don't eat your nucleic acids, but you do eat them. That's not what you use them for. But if you don't eat the stuff, you can't make the stuff. So you have to eat what your body needs to create you. And if you don't, you're going to be malnourished in some way. So all of these organic molecules, the shape determines the function. I mean, they're all very, that's true for all of them, but proteins are super, super specific. So we're going to kind of draw some here in just a minute. The functioning shape of a protein, when it's functioning right and it's made correctly and it's doing what it's supposed to do, it's called its native state. So I'm sure you've heard of denatured proteins before. If you've ever watched an egg fry, you've watched it happen. You've watched that white turn from being very watery and liquidy into, almost a solid. And that's because the proteins of that egg white became denatured and they broke apart and they no longer functioned correctly. Same thing can happen with us. a change in our temperature. The reason you don't want a high fever for a very long period of time, or you don't want one at all, but is because it can denature our proteins. So what I've always taught is that around the temperature 102 is when you need to, make sure you start bringing it down. Don't be a parent that gives your kids Tylenol for fever at, 99 or 100, unless they've had, ongoing increasing fevers. Let your body do what it's supposed to do. You're supposed to have fever. You're supposed to have a little bit of a fever to try to, kill off whatever is causing this fever. So allow your children to have a little bit of fever. It makes you feel terrible, and that's on purpose. When you are sick, you shouldn't feel great. So you should, chill out and watch TV. But if you change the temperature too high, just like the egg, you start to denature proteins, and so they lose their they can't function correctly anymore. So that's definitely 102 is kind of where you need to make sure you kind of get it brought down. Change in pH, radiation, chemicals, drugs, all that kind of stuff, other things can denature proteins. You're just breaking either weak hydrogen bonds or you're breaking covalent bonds, depending if it's extreme enough. So, but when they're working correctly, they're said to be in their native state. So we learned the very first level of protein structure in biology. So all of the protein synthesis stuff that we learned in biology was just to get the very first structure done. So the primary structure is that released polypeptide chain. Now I'm just going to use one color because I'm going to use purple throughout. But we talked about how the ribosome reads the mRNA and the tRNA brings in the amino acids. So that primary structure is just a chain of amino acids just linked together in the specific order that the DNA said to be linked together. And then it's released. Okay? So we call this a polypeptide chain. Because the peptide bond is what holds these amino acids together, so it's many peptide bonds. So hundreds of amino acids linked together in a specific order create just the very first structure. And like I said, in biology, we learned that was it, and that's untrue. Okay, so the secondary structure, remember that you are an ongoing chemical reaction, just all the time going on, and trying, all these elements need to be as stable as possible. So these amino acids have bonding availability. They still have some of their electrons or their orbitals are not full or whatever, so they need to, share or do something. They need to make some bonds. So the secondary structure is where that amino acid chain coils up onto itself, and I'm not going to draw the bunch of dots, but that's the dots coiling up onto itself, or it creates sheets, pleated sheets. So it folds, somehow creating these bonds that are making it stronger, making it more stable. So the secondary structure is to get as stable as it can through weak hydrogen bonds by itself. Okay, that's the secondary structure. Oh, hold up, my camera's stuck. Sorry. Okay, the tertiary structure is where the coils and the pleats and stuff start to make stronger, maybe covalent bonds, and it folds up even further onto itself and just makes stronger bonds to get as stable as it can. Now some proteins, this is it. They might get to tertiary and they might be stable, and that's if that's what they're supposed to do, then that's great. Others go ahead and go into this final quaternary structure, which is you see these used all the time in movies. I was telling my students about, you see them used, and they use them as like viruses and stuff like that, and usually it's just a protein because they look kind of crazy. So multiple quaternary structures come together and get stable, and they form that functioning native state protein. So I'm just going to redraw my purple one here, sort of. And then I'm going to link it with, get a couple of other colors. And they don't all coil in and fold, but so they're a very specific structure, and they intertwine and they just connect all throughout themselves and each other to get as stable as they can. And it seems random, but it's very much not. You've got polar and non-polar, kind of trying to get away from water or toward water, or hydrophobic and hydrophilic. You've got different parts, and these become specifically shaped so that they can do their job. So the inappropriate folding, if something, if we go all the way back to DNA mutations and there is a G instead of a T, then that is going to code in the RNA incorrectly. And luckily, quite a bit of codons repeat, and so you could get the same amino acid for 4 different codons. But it just depends on if that brings in the right amino acid as to if this is going to fold correctly. So you have to go all the way back to DNA if this is incorrect. So sometimes we're lactose intolerant. We don't make lactase enzyme right. It's not going to kill us, but that's what we carry in our genes. That's what our DNA says. So we can make the enzyme, but it does not break down lactose sugar. So we can pass that on to our kids now because it's not going to kill us, so you just pass it on to your kids. So more and more people can be lactose intolerant and survive it because we can survive a mutation like that. Okay, so there are 2 major types of proteins. Structural, which are going to be the ones that form the structures of your cells, whether it be inside the cell somewhere or if it's the protein channels that are embedded in your cell membrane. Different tissues and organs of the body are very, very, condensed protein structures, like tendons and ligaments are very fibrous, thread-like proteins that are strong. And so those are structural examples of proteins. Functional examples are more like enzymes. So the enzyme is a functioning protein that helps to actually speed up our chemical reactions. And these are the chemists of our bodies. They don't make things happen that can't happen without them, but they do speed them up to a point where we can live. Because if we just I call these the farmersonly.com or the whatever they're called, all those, find love on the internet kind of places. You could meet that same exact person just randomly somewhere throughout the world, but the chances of you meeting them are a lot better if you're both on that same website. So that's what an enzyme is. It's a place that makes chemical reactions proceed at a rate that keeps us alive, and it makes it speed up so much faster than if they just happen to break apart or they just happen to come together. So functional enzymes have a very unique shape that allow them to fit certain chemicals and cause specific change in the molecule. Sometimes they cause catabolism, which is breaking down. Sometimes they cause anabolism, building something together, and sometimes they can do both. But enzymes, hormones are an example of a protein that, we all know for sure that hormones make things happen. So that make specific changes happen when they're released. Antibodies that help fight off invading infections and stuff. So enzymes are specific, and you think of these kind of like a lock and key. If they're not shaped right, it's just not going to happen. I have a whole set of school keys, and they all look alike, but only one opens this door in my classroom. Others look just like it, seem to look just like it, but they're just a little bit different. So they don't actually open that door. They open something else. So if it's made just a little bit wrong, it's not going to work. So with an enzyme, let's just draw of course this is super simplified, but I'm going to draw a triangle and then a little gap and then like a rectangle. So let's say that this is an enzyme to break down sucrose, which is table sugar. So sucrose comes along and it's shaped like this. So it can fit in there just like a puzzle piece. It can just fit right in there. And this is called the substrate, whatever it is that's supposed to fit into the enzyme. And then this is your enzyme. And then this right in here is your active site. So these are left, these are bonds that are left unstable. So when these come in and they fit in here, then those bonds are made, and then whatever this enzyme changes shapes can break these two pieces apart and release them as glucose and fructose or whatever it is that's supposed to be broken apart. It can also take something that's shaped like a triangle and something that's shaped like a rectangle or square, and when they both dock, it can put them together. It can twist and basically put them together. So enzymes just make chemical reactions happen at a rate that keep us alive. Okay, so that's all we're going to learn about proteins, and the next one is going to be lipids.